you're going to end up producing at the end of beta oxidation, you're going to be making acetyl-CoA, you're going to be making NADHs, you're going to be making FADH2s, as well as eventually generate ATP. But we'll talk about this one in a minute. Right now, we're going to focus on how we break down those triglycerides. So how we break it down is another lipase. This one is hormone-sensitive lipase. That's what's going to cleave the fatty acid off the triglycerides. So when we're taking off the fatty acids, that's a lipase. When we're putting on fatty acid, that's a what? Acyltransferase. So in this case, we're taking them off, that's a lipase. And the lipase, specific lipase, is called hormone sensitive. It's going to cleave the fatty acid off the triglyceride, eventually to produce the free fatty acids as well as the free glycerol. And it is regulated. So this is another regulated enzyme. Woo, I know you love those. I try to limit them, and I just want to give you a heads up. All the enzymes are probably regulated somewhat. I'm focusing you on the major regulatory enzymes. It's not necessarily black or white, but I am focusing on the major regulated ones. And then this one, I'm going to keep it simple. It's activated. You already told me what it's activated by. It's activated by glucagon and your stress hormone epinephrine. So epinephrine is one of your catecholamines, epinephrine, norepinephrine. These are your stress hormones. So they come out when you're running from a bear, taking an exam, you take your pick. But these guys, glucagon as well, they'll release fatty acids. Essentially, you need fuel because you're in the fasted state or in the stress state. So if you're running from a bear, you also need fuel. And it's going to be inactivated by insulin, which remember has a phosphatase. It's going to take off the phosphate group. So this one, pretty simple. I'm not talking about any allosteric regulation for this one, but do know the covalent regulation. And this is just a little bit more on the hormones. So again, hormone-sensitive lipase is activated. It's going to allow the mobilization of fatty acids. So mobilization means they're being freed for use. So the mobilization of fatty acids from fatty tissue, adipocytes, these hormones are going to activate hormone-sensitive lipase. The ones I do want you to know are glucagon, epinephrine, norepinephrine, and cortisol. Norepinephrine, that's what NE stands for. It's norepinephrine or norepinephrine. And I usually just call it NE. So do a lot of other people. Common acronym. And epinephrine is EP. These guys, and cortisol. So epinephrine and norepinephrine, the general population, do you guys know what they call this guy? Adrenaline. So whenever you hear someone talk about adrenaline, they're actually talking about epinephrine. And noradrenaline is norepinephrine, not that we really say noradrenaline. But they got their name, they're released as hormones from the adrenal gland. So we do call these adrenaline, but adrenaline is actually epinephrine and norepinephrine. And they have very similar functions, so we tend to use them interchangeably. They're also neurotransmitters. So technically norepinephrine is more of a neurotransmitter, epinephrine is more of a hormone. But they do the same things. They increase your blood flow, increase blood pressure, and they actually help you break down fuel so you can have more energy. There's a question, yes?
Is that the same thing they give people who do a drug overdose?
No. Actually, I have that in my cabinet, so I'm trying to remember what that is. Narcan, that's, my dad has a lot of issues. We have to have stuff prepared just for him. But you're also thinking about the EpiPen. The EpiPen, when you have an allergic reaction, I teach physiology. You guys might have me in a couple terms for that. But EpiPen is one of those, if you have allergic reaction, a really strong one, that essentially can constrict your air passageways. And so epinephrine, if you have a shot of epinephrine, remember it's a stress hormone / neurotransmitter. So what it's going to do is open your air pathways, and so if you're running, the body's assuming it's scared, and so you want to run away, and so it's going to allow you to breathe in more oxygen so you can run away better. And so that's why the EpiPen, yeah, that's it. I love physiology. I won't go more into that though. We can wait for a couple of terms for you guys. But keep in mind, it's the ratio of hormones. It's not like insulin goes away. It's just insulin at this point, there's very little insulin. So glucagon in this state is going to be the primary controller. But these three are the ones you probably should know are going to trigger lipolysis. The others do it too, but I don't usually talk about them as much. You'll probably get them in physiology some more. But we haven't talked about them in this class, so I'm going to kind of ignore them for now. And this is the figure from your textbook. So again, it's saying the same thing. It's just showing you the figure as well. And I also want to point out, because we talked about this before, but sometimes I feel like it's, I want to make it clear. Insulin doesn't phosphorylate or dephosphorylate anything. It sits on its receptor and it triggers a signaling cascade. And that signaling cascade will activate a phosphatase, which will take off the phosphate group, rendering hormone-sensitive lipase inactive. Whereas epinephrine, or this could just as easily be glucagon, also will just sit on a receptor, and it's going to activate a kinase through that signaling cascade that's going to then phosphorylate hormone-sensitive lipase, which makes it active. So keep in mind, insulin and glucagon don't personally, on their own, phosphorylate or dephosphorylate. They trigger a signaling cascade that does it for them. But that's the covalent regulation of hormone-sensitive lipase. All right. Release the fatty acids from adipose tissue. So here we go. Glucagon will bind to its receptor. It'll start a signaling cascade. Technically, it's going to activate adenylate cyclase, which is going to phosphorylate AMP to make cyclic AMP, which is what's going to activate protein kinase, which is what's going to phosphorylate hormone-sensitive lipase. Don't memorize all those terms. I just wanted to walk you through the signaling cascade. This is the signaling cascade. All you need to know is that glucagon is going to start a signaling cascade, which is all this, that's going to activate a kinase, which is going to phosphorylate hormone-sensitive lipase. Here, active because it's phosphorylated. That's the part you need to know. So you don't have to memorize this stuff if you don't want to. But you do need to remember that it activates a kinase, which will phosphorylate this hormone, make it active, and when it's active, it will pull the fatty acids off of triglycerides. Hormone-sensitive lipase will pull the fatty acids off the triglycerides in order to make free glycerol and free fatty acids. And this is in tiny words down here. Well, not really tiny. But just so you know, remember I told you that it's really hard to pull off that middle fatty acid from the glycerol? So during digestion, you have the glycerol group and the fatty acids. And it's really hard to actually tear off this middle fatty acid. So technically, there is a different enzyme that's going to cleave the last fatty acid. Don't memorize that enzyme, but that's what this is saying down here. It does need help to remove that last fatty acid. Yeah. Yep. So the glucagon signals cascade active that phosphorylates the hormone-sensitive lipase. That phosphorylates the hormone-sensitive lipase. Yeah. It takes off the fatty acids from the triglyceride. So lipases take fatty acids off things. Acyltransferases put fatty acids on things. So whenever you see a lipase, because there's lots of lipases, there's lingual lipase, there's lipoprotein lipase, there's hormone-sensitive lipase. Every time you see a lipase, just try to keep this in your memory bank. That's taking fatty acids off things, away, taking them off. It cleaves them. An acyl transferase, keep this in your memory bank too. Acyl transferases put fatty acids onto things. So lipases cleave them, take them off, and an acyl transferases add them. It puts them onto things. And I'll say this 100 times, so please don't ever feel bad if you're asking a question. I will. Repetition is so important for learning this language. So the fate of the triglycerides, you could also say they're hydrolyzed. Know that hydrolyzed means break down. When I see hydrolyzed, I already know, oh, something's being broken down. So whenever you see hydrolyzed, it's being broken down, and it's being broken down with water. So otherwise, you wouldn't be able to say hydrolyzed, but technically it's with water, so that's why it's being, why we call it hydrolyzed. We could say catabolized, which isn't very specific, but also means something's being broken down. But that's what happens. Triglycerides are hydrolyzed, broken down into free fatty acids and glycerol. Glycerol can be used to synthesize new triglycerides. Apparently I have to write this 3 times in the slides. But it must first travel back to the liver to make glycerol 3-phosphate first, which we just talked about that. And that's, I feel like I have a duplicate slide. So adipocytes lack the enzyme glycerol kinase, and so you can't add a phosphate group. You need that glycerol phosphate, 3-phosphate, in order to make a new triglyceride. So free fatty acids, why do we free them? We free them primarily so they can be oxidized. Oxidized means we take away their electrons, and those electrons are put on the electron carriers, NADH and FADH2, for the electron transport chain, and then in the electron transport chain, we're going to make ATP with them. So fatty acids are oxidized, and that is called beta oxidation. We're still not doing beta oxidation yet, but we are so close. But just keep in mind, the brain does not use fatty acids. Most of your tissues can use fatty acids. The only 2 tissues that does not use fatty acids are your brain and your red blood cells. Those are the 2 primary tissues. The brain doesn't use them. It only uses glucose or ketones. And part of that is to transport. The brain is highly protected, so it has mitochondria, but it doesn't use the fatty acids for energy. It can only use glucose or ketones. Those are the 2 smaller ones that can come and enter the brain. Red blood cells don't use fatty acids. Why? Do you know that? Do you know why the red blood cells cannot use fatty acids for energy?
They don't have mitochondria.
They do not have mitochondria. And to do the Krebs cycle, the electron transport chain, all that stuff, you need a mitochondria. And so red blood cells don't use it for that reason. So fatty acids can be oxidized for energy except for red blood cells and the brain. All right, we're almost getting to beta oxidation, but now first we have to get the fatty acid to the cells. So how do they get to the cells? When they're freed, they actually bind to albumin in the blood. There's no lipoprotein that can carry them in this pathway. So with lypolysis, when you get that free fatty acid, it's just going to enter the blood, bound, and bind itself to albumin. And keep in mind, this might limit the amount of free fatty acids because you can't put them in the blood unless you bind them to albumin. So if albumin, the concentration of albumin decreases and there's no more transport, then that will also limit the ability of hormone-sensitive lipase. So hormone-sensitive lipase will not work as well when all the albumin is taken up. So if there's no more free albumin, then hormone-sensitive lipase will decrease its activity. because why pull off more fatty acids if they can't be carried to the tissues? Okay, so now we're getting closer to doing beta oxidation, and this is telling you what we're going to make with beta oxidation. The goal of beta oxidation is going to be to make acetyl-CoA, NADH, and FADH2. And all these guys you recognize, and that's actually what we're going to make with beta oxidation. Which I guess we're not talking about yet. So here's the free fatty acid bound to albumin. So we're in the blood. Now we got to get this guy into the cell. There's a transport mechanism. And I've decided I'm not going to make you guys memorize this because I don't think it's going to be something that you would ask about on the boards. So there is a transport mechanism for getting these guys into the cell, but don't memorize that. So once they get into the cell, though, there's free fatty acids. This is what you do need to know. What's going to happen is that fatty acid has to be activated. Fatty acids cannot be used in the body unless a coenzyme A is added to them. They just can't be used. So you can have the fatty acid there, but it's not going to be used unless a coenzyme A is added to it. So as soon as it enters, a coenzyme A is going to be added to it, and that's going to, we actually call it activation. We activate the fatty acid by adding a CoA to it. So here's the CoA, the fatty acyl-CoA, and that's added using this CoA, acyl-CoA synthetase. This is not a really important enzyme. I definitely won't ask you about this one. But it's the enzyme that's going to add the coenzyme A to the fatty acid. So now we have fatty acyl-CoA. Fatty acyl-CoA, it's now ready. So you can store it, and so sometimes the muscles might store it. That INTG just stands for intramuscular triglyceride. But most cells are going to use that fatty acid for beta oxidation. So now we're going to do beta oxidation, but keep in mind the fatty acyl-CoA cannot cross the mitochondria. So that's where this guy comes in. This guy is carnitine. Carnitine is going to be able to transport the fatty acid into the mitochondria. Yeah.
So,
Yeah, the free fatty acid detaches, it goes into the cell, and then the coenzyme A is added. So it's actually two separate processes. It leaves the albumin in the blood, and it detaches from it, binds to this receptor here, and it binds to this receptor here, and comes in, and then the CoA is added. No, don't memorize these receptors, but do know that I just want you to understand it can't just go into the cell. It will need a transporter. But don't memorize the transporters, but it will need a transporter to get into the cell. And then once it's in the cell, that's when the coenzyme A is added. And now it's officially active. It can be used, in other words. So now we got to get it into the mitochondria because that's where beta oxidation happens. Where does fatty acid synthesis happen? In the mitochondria or the cytosol? Cytosol. So we separate, your cells separate fatty acid synthesis from fatty acid breakdown, which is good. Fatty acid breakdown or beta oxidation happens in the mitochondria. Fatty acid synthesis happens in the cytosol. So they're happening in two different places in the cell, so the cell is able to regulate them more easily. All right. In the cell, again, I like to repeat myself. In the cell, fatty acid is activated by coenzyme A, and again, here's that enzyme, acyl-CoA synthetase. It is a good one to know, although I don't have this one on the exam. Oxidation of fatty acid occurs in the mitochondria, and that's going to allow us to produce ATP. But first, we have to transport the fatty acid into the inner mitochondrial membrane. And so carnitine is a really important molecule. We actually can make it I know that there are supplements out there, carnitine supplements. Generally speaking, a healthy person is not going to get anything out of those carnitine supplements. They're really for people who have, either malabsorption issues or are not properly getting enough protein in or various other reasons, but our bodies make it. It makes it from the amino acids, glycine and methionine. But you actually don't have to memorize that yet, although we are going to talk about amino acids later. So if you have a short to medium chain, it can cross the mitochondrial membrane without transport. So this carnitine is only for long chain, over 12 carbons. So if there's more than 12 carbons, it will require carnitine. But if it's a little one, then it can pass through without a problem. And there's two enzymes, and they're really easy to remember because they're actually called carnitine acyl transferase. So acyl is the fatty acid, transferase is what they're doing into the mitochondria. Yeah. So the reason why short and medium chains can pass through is because they're small enough. And they're lipids. And the mitochondria is a lipid membrane. And lipids like lipids, and so they just allow them to pass because they're small enough. If a lipid is really big, it can't pass through the membrane. So that's why the short ones can do it, because they're lipids and they're small enough. But yeah, the really big ones, even though they're lipid, they're just too big to get through. But yeah, that's a good question. So carnitine acyl transferase 1 and carnitine acyl transferase 2. Remember, there's two membranes for the mitochondria for the electron transport chain and all that stuff. So you have to move it first through the first membrane. That's going to be the carnitine acyl transferase 1. And then to get it into the inside, the very middle of the mitochondria, you have to pass it through another membrane. And that's going to require, I just see a misspelling here, carnitine, not canitine. Carnitine acyl transferase. So many typos in this lecture. I thought I caught them all. I clearly did not. So two enzymes facilitate fatty acid transport. Carnitine acyl transferase 1 moves the acyl carnitine across the membrane. So I should mention here that what's going to happen, you're going to see this in the slides, but you're going to have, so here's the mitochondria here, and here's the fatty acid, which has the CoA attached. To get it into the mitochondria, all right, I'm going to try to do this. I'm so going to do this, and it's going to get great. So we're in the cell. Here's the fatty acid, CoA, and we're going into the mitochondria here. And remember, it's a double membrane. The mitochondria is a double membrane organelle. And so we have this fatty acid CoA. In order to get in here, we need this carnitine, which is going to be sitting in the membrane here, this carnitine acyl transferase 1. And what it's going to do is it's going to take this fatty acid CoA, and it's going to take off the CoA, and it's going to add carnitine to it. That's why it's fatty acid carnitine. So it's actually, when it comes into the cell, it's going to be fatty acid carnitine. It's going to be attached, instead of to coenzyme A, it's going to be attached to carnitine. So that's where carnitine comes in, because you're going to have to add a carnitine to it. And that's what CAT1, or carnitine acyl transferase 1, is going to help us do. So it catalyzes the conversion of acyl-CoA into acyl carnitine. So it takes off the CoA and it adds a carnitine to it to carry it across the mitochondria membrane. So you just switched out the CoA for a carnitine. And we're going to maintain the carnitine in order to get it through acyl transferase, or carnitine, I did the same thing here, carnitine acyl transferase 2. So it's going to bring the acyl carnitine in, and it's going to release the carnitine and add back the CoA. So carnitine acyltransferase 1 gets rid of the CoA, adds a carnitine. Carnitine acyltransferase 2 is going to get rid of the carnitine and add back the CoA. So ultimately, at the end, we're back to an acyl-CoA, which is another way of just saying fatty acid CoA. They just call it acyl-CoA. So either way works. Acyl-CoA and fatty acid CoA, same thing. Is there a question over here? Yeah. Oh, you want to? You like that? It's so good. I'll tell you that the other one is going to be in this membrane. So you see Cat1 here. Cat2, carnitine acyltransferase 2, is going to be here. So this will be carnitine acyltransferase 2 will actually be in the inner membrane. So Cat1 is right here in this first membrane, and Cat2 is here in this middle membrane. And so that will bring us here. We're going to be able to bring in the fatty acid, and we're going to add a CoA back to it. And now we have fatty acid CoA in this middle of the mitochondria here. That's where we want to put it in order to do beta oxidation. Yeah. It's actually in the membrane. I tried to put it in the membrane here. It's actually in the membrane. I have it looking like it's outside, but it's technically inside. I have another picture of this that might be better than my drawing. I don't know. My drawing's pretty nice though. So, let me just, here you go. Better picture. All right. But here again is the mitochondria, so that's where we're trying to get the fatty acid in here, in the middle of this mitochondria here. And so, what we're going to do, here we're in the matrix, we're in the cytosol on this side, and we're going to take this fatty acid CoA here, this is the fatty acid CoA, we're going to get rid of the CoA, so the CoA is going to be dropped off, and we're going to add a carnitine to it. And that is going to allow us to transport into the intermembrane space, and then, I am going the wrong way, aren't I? Yeah, never mind. The matrix, sorry, I just went the wrong way. Oh, and it won't let me erase now. Yeah. Maybe not that one. All right, sorry guys, can I back up? Realize, can I do a refresh? Go back like 30 seconds. Let's pretend I didn't talk for the last 30 seconds, and we're going to start in. Oh, you guys need a break too. All right, we're just going to take a break, and I'm going to get back to this. Quickly first, and then I'm going to get back to this. Oh, mine's not working. Oh, there it is.
What rhymes with barley? I said barley, yeah, iCarly. What? Yeah, barley.
Can I do it backwards?
Were you rapping the parley right now? Earlier. You better not be rapping the parley without me. What's up, man? Or what?
Yeah, I mean, like, Or, actually, I started that when I started. But yeah, I'm almost I never really noticed that before. But I never really thought about that before. But yeah, this essentially, in order, so these are actually just short pieces of the enzyme that are not active. They literally have to be, I don't know, like I want to know this, they have to be put together like that in order for it to pass into it, which apparently only happens
straight down.
Daddy, what you working on?
Something that makes more sense.
So your mom too?
Any kid whose parents is a teacher, that academic is crazy. They know the system.
You're academic, right?
She's been an academic. Yeah, she over there always answering
Her dad
questions.
is that.
Oh, for real?
Yeah. I just found that out. It's like off the color.
I'm saying.
She was right next to she was talking about us before.
I'm saying.
She was playing dog.
He said, pause for the takes.
God, it's okay. How you doing, man? I'm about to quit.
Already?
Yes. I need more time to do it. I want more time to do school stuff, to go to clubs.
When you just take this out.
I'm going to work for my dad instead. I'm not like,
It's ownership, bro.
so my dad's entrepreneur. It's just hard with one.
That's gonna drain you.
I've been doing this for a decade.
Did they lock me?
But it's because the hours, my dad was way less hours. Right now, I'm doing like 21 to 22 hours of work in a week. Big work week. My dad might work like 12 to 14 and make twice as much money. So,
Why did you have to start?
you know, because I was rebelling. But obviously, I didn't go back.
You're gonna come back home, don't worry. Pops gonna be like, well. You always come strong back.
Mom's to it, like, but that's still really good.
We'll talk about the cameras.
Yeah, I mean, it's usually by carbon
What we talking about?
But yeah, you just need the carbon. So either one bicarbonate. Looking at it here. So looking at it here, all at the light is concentrate there. So I have put that on. Yes. So I'm wondering if that's back there.
Was he there?
Yeah,
So look for that. But it is like you start with the super way, you got it. You get it. And that's going to that is so it's really it's everything. So this really is everything essentially that you need to know. So you don't have to draw it all out. But yeah, this is what you need to know. I have it in a written form like that. But this other big picture, I don't like it because I think this one, if it's on the slides, like, yeah, these are the That's how it goes.
This one, citrate is away. Yes, it's a shuttle that takes everything away. I mean, if you look for yeah, for first step, we're going to reduce alcohol. First step in finance for business is to make alcohol. I really feel that. Like I would say, I would say, away. And I have to see all there's it's like, this is what we did today. But I am not worried about that. I think that's, there might be some side. I don't worry about especially because yeah, so it's depending on the type of learner you want. So what I would do so I would make sure this is it. You just need to make sure you're adding the right enzyme. So this is so yeah, this one make sure there you don't really have right enzymes. That's what I want to focus on. Like that's what it is. You do have the acetyl-CoA, and you have it making the malonyl-CoA. Now the enzyme, we can go back to that. Then you take malonyl-CoA, and there's malonyl-CoA, and you add like the acetyl-CoA to it, and that enzyme is going to be so these, you're going you're going into way more detail. That's what I want to say. Like you're actually following more this, which is not bad. Like you're actually looking at it from all the steps. What I want you to focus on is the big picture of once you have, so the first step, once you make your malanote away, and that happens step one is you make the malanote away. And that was what you had here with, no, that's not that, it's the one up there. So this is, yes, I think that's why I'm confused, because you have all these. So this is step one. Yeah, this is step one, and once you have this, then step two is this. And the enzyme that does it, it's fatty acid synthase. So fatty acid synthase has this acetyl-CoA attached to it. It adds the malonate away. And when it does that, every time it does, just draw it out. Yes. So I like to think of it this way. I don't draw like Dave was drawing it all out. Yeah. Not even think. It's just the first step it's going to be you take a 2-carbon, which is acetyl-CoA. And you're going to add a carbon to it. And usually it's bicarbonate, HCO3 minus. And that's what you make a 3-carbon malate. And that's acetyl-CoA. Same thing on acetyl-CoA carboxylase. That's the enzyme that's going to do this. And it makes sense because carboxylate means you're carboxylating something. And you're taking a 2-carbon and you're adding a carbon to it. So now you have a 3-carbon malate. Exactly. And so in step 2, you're going to have, that's when this malanote away, so you're going to have, the enzyme is going to be fatty acid synthase. This is the enzyme that's going to do it. And it's going to take one acetyl away, and plus a malanote away. Okay, can I just get this wrong? Hey, sorry. Yeah, I'll see you at 3. So the 2-carbon acetyl-CoA is going to condense with a 3-carbon malonyl-CoA. And what you said just a minute ago was exactly right. It's actually going to take off. It's actually going to take off one of those carbons. And what you end up with, and by the way, this is what fatty acids make this reaction. And what you're going to end up with is a 4-carbon, 1, 2, 3. Because you lose one of those carbons in the reaction. And so that is step 2. So step 1 is where you take the acetyl-CoA to make a malonyl-CoA. Step 2 is where you take that acetyl-CoA to take another acetyl-CoA and add the malonyl-CoA that you made in the first step. You're going to do that. This is what you're going to repeat over and over again. Fatty acid-CoA, so you're going to take this guy, and it's going to add another malonyl-CoA, another 3-carbon malonyl-CoA. Yep, and it's going to do the 7 times, so we're going to lose another carbon, and that's where we end up with 6 carbon. 16 carbon, 18 carbon, 20 carbon. You can make upwards of 24.
Said that was gonna like I almost wanted to just like see
I'm so well, I'm not like a like I was. I I still would run it occasionally. Like I want to wear like a flip flop. Alright guys, I'm gonna do the figure correctly this time. It is correct on the lecture video. My brain just, whatever it does. But this picture is actually a blow up of the mitochondria. So it's kind of looking at it from here. And I think I wanted to get it into the mitochondria, so my brain went the opposite direction. Okay. So now we're in the cytosol, not the matrix. The cytosol of the cell. And we have to get this fatty acid OA, here it is, into the mitochondria. So this represents the fatty acid. Every time you see an R, it just represents a group. In this case, if we're talking about fatty acids, it's representing the fatty acid. In the amino acids, last term, the R represented the different groups of the amino acids. So whenever you see an R, it's not a molecule itself, it is representing a molecule. In this case, it's representing a long fatty acid shell. And we don't want to have to write out the fatty acid tail every time, so it's easier to just put R. So in this case, this is the fatty acid. Notice that we've attached it to a coenzyme A. That's how we activate it. So now we can use it. The cell doesn't recognize it unless has a coenzyme A added to it. And so now we can, what carnitine acyltransferase is doing is it's going to actually take off the coenzyme A. So here's the coenzyme A leaving, and we're going to attach instead a carnitine to it. So here's the carnitine, and now we're going to put this carnitine onto the fatty acid. So that's what's going on here, and that is done by this CAT1. The carnitine acyltransferase 1 is what is doing that. So there it is, carnitine acyltransferase 1. And that's just getting it into the inner membrane space. We got to get it into the matrix, the very middle of the mitochondria, which means we've got to now move it into or through this other membrane. So now we've got this membrane we got to move it through. And so we keep the carnitine on it, and that's what carnitine acyltransferase 2, or CAT2, will help bring in this guy. So it's a double enzyme, by the way. It acts as a transporter and an enzyme. So it's going to transport this guy in and take off the carnitine and add back the coenzyme A. So we're back where we started. So here's the same fatty acid as it was out here. It's the same fatty acid. Now we've just got the coenzyme A added to it. Now we're ready for beta oxidation. Because we have the fatty acid in the cell, or in the mitochondria, and it is attached to a coenzyme A. Now we can use it for beta oxidation. But there is one regulation you should be aware of when it comes to transferases. I'm going to get a question first. Do I need everybody to solve this? Yeah, no worries. I can always go back to this and redraw it. So again, the key is make sure coenzyme A isn't on it to make it active. But if you're transporting the mitochondria, you have to take off the coenzyme A and put a carnitine on it. But that's just for transport. You just need the carnitine to transport it in, and then you add back the coenzyme A, and now we're ready to roll to do beta oxidation. But keep in mind this CAT1, the acyltransferase 1, is regulated by malonyl-CoA. Where did we see malonyl-CoA? Fatty acid synthesis. Yeah. Nope, it's just moving in. So nothing happens to it between the carnitine acyltransferase 1 and carnitine acyltransferase 2. They're just, we need two of them because we have to go through two membranes. So nothing happens to it though in between. It's not until it gets to the carnitine acyltransferase 2, that's the one that's going to take off the carnitine and put the CoA back on. inside the mitochondria, in the very middle part, which is the matrix of the mitochondria. Yeah.
That's it. Up. Right there. Right there. Right,
Yes.
right there. Come on. Come on. Why does yours look different than mine?
So malonyl-CoA does not, it might, if it does, it's not something that we really, that you need to know. So
Whatever basic thing.
I guess there's gray area, always gray area, but generally no. So it doesn't do anything, well, we use it, it does do a lot. We use it for fatty acid synthesis, so that's where we've seen it before, is malonyl-CoA. It's the very first step. So step 1 of fatty acid synthesis, we make a malonyl-CoA. Let me ask you guys, does it make sense for the cell to do fatty acid synthesis and fatty acid breakdown at the same time? No. That's what this is doing. You do not want fatty acid synthesis and fatty acid breakdown happening at the same time. So if you're doing fatty acid synthesis, you're making a lot of malonyl-CoA. If you're doing fatty acid synthesis, you're making a lot of malonyl-CoA in the cytosol. So it's already floating around in the cytosol, and if you have too much of it, or a decent amount of it, it will inhibit carnitine acyltransferase. And that prevents beta oxidation and fatty acid synthesis, which are the opposites of each other. It prevents them from happening at the same time. So that's the only regulation you need to know for this one, carnitine acyltransferase 1. And a lot of other enzymes too, yeah. But it's not like it's, for gluconeogenesis and glycolysis, that fructose 1,6-bisphosphate or 2,6-bisphosphate, yes, that is very similar. It's the same type of regulation. So. Not yet. All right, so here is from your textbook. It's the same picture. I just liked mine a little bit better, so I put mine in there. But the cytosol is where you have the fatty acid CoA. You have to add the CoA to make it active. When you bring it into the cell, you have this carnitine acyltransferase. And they sometimes, the textbook called it carnitine palmitoyl transferase because you're usually using palmitate. 16-carbon palmitate is usually the substrate, usually what the cell's using. But again, I prefer acyltransferase because you don't have to use palmitoyl palmitate. But you get it into the mitochondrial matrix, and that's when you have the fatty acid back again attached to back again attached to coenzyme A. So the carnitine is removed, and you need to remove the carnitine as well because carnitine has to go back out and pick up more fatty acids. So carnitine is just being attached to the fatty acid temporarily just to get it into the mitochondria. Carnitine, we get it from the diet. It is in our diet. It is primarily in meat products. Your muscles will have a lot of carnitine because muscles will be using a lot of fatty acids to make ATP. So muscles have a lot of carnitine, so if you eat meat, it will be part of your diet, but it can be synthesized. So it is not essential. We can make it. You can be a vegetarian or even a vegan and make your own, and your cells can make your carnitine from amino acids. So the carnitine shuttle, and the carnitine shuttle is inhibited, like I just said, by malonyl-CoA, which is present in the cytosol during fatty acid synthesis. And it is true, we can buy supplements for carnitine. If you don't have enough carnitine, you can't break down LCFA. So LCFA just stands for long chain fatty acid. Long chain fatty acid. So people with liver disease or malnutrition, but some vegetarians, people who are pregnant, other patients, infections, burns, anybody who's got other deficiencies may benefit from a supplement. But usually most healthy individuals don't need to supplement. Yeah.
What is the primary difference between creatine and carnitine?
Creatine and carnitine. So creatine is creatine phosphate. Creatine, I used to get those mixed up all the time, so it makes me laugh. But creatine, it's used to provide ATP fast. So in the muscles, your muscles have a lot of creatine. It holds onto a phosphate group that can be given to ADP to make ATP fast. So if I were running away from a bear, my muscles would use creatine phosphate first to make ATP, because you can make ATP really fast with creatine phosphate. So creatine is all about making ATP fast, whereas carnitine is about just fatty acid transport. Yep. And it's so easy to get those confused, or at least it was for me. Now we can do beta oxidation. It feels like I've been talking about beta oxidation, but we haven't started it yet, but now we can. Beta oxidation is a degradative, a breakdown pathway where 2 carbon units, acetyl-CoA, keeps popping up when it comes to lipids, 2 carbon units, acetyl-CoA, are cleaved at the carboxyl end one at a time. It occurs in the mitochondria. And again, just like fatty acid synthesis, the steps are repeated. So you're going to keep doing this until you cleave off 2 carbons at a time to make acetyl-CoA from that fatty acid. So it really is very similar to the fatty acid synthesis. Instead of adding 2 carbons at a time, you're breaking off 2 carbons at a time. And why do we call it beta oxidation? It's called beta oxidation because the names of the carbons, you guys don't have to memorize all this, but technically this carbon here, the one that's attached to the carboxylic acid, that's an alpha carbon. The second carbon is, beside the alpha carbon, it's always the beta carbon, alpha, beta. And the cleavage happens at the beta carbon. So that's where you have cleavage. That's really doing a good job there. That's where you have cleavage, is at the beta carbon, and that's why we call it beta oxidation. Do you need to memorize that? No. I just want you to know why it's called beta oxidation. So it has to do with the positioning of the carbon that is cleaved. So the beta carbon is the second carbon in the fatty acid chain, counting from the carboxyl end, and the beta carbon is involved in the cleavage. So that's why we call it beta oxidation. So when we cleave that, and it's a thiolase is the name of the enzyme that's going to do the cleaving. It's going to cleave between the alpha and beta carbons to make a 2-carbon acetyl-CoA. So every time we take off 2 carbons, we're actually making an acetyl-CoA, and I'll show you what that looks like. So every time the fatty acid is cleaved, it's shortened by 2 carbons. So here's, well, I'll just go through it first. It's pretty simple. These are going to be all the steps that I'm going to have you guys learn. It looks like a lot, but it's not. Acyl-CoA dehydrogenase is the first step. Dehydrogenase. You might remember this from last term. What do we make when we have a dehydrogenase? We either make an NADH or an FADH2. In this case, we're going to make an FADH2. But that's dehydrogenases tell you we're going to make an FADH2. We're going to make an electron carrier. So the first step, we're going to make an electron carrier. And we actually have different types of acyl-CoA dehydrogenases. For the very long chain, for the long chain, medium chain, and short chain. They all have their own acyl-CoA dehydrogenase. So when you start with a very long chain, let's start with a 24 chain. When you start splitting that down to like an 18 chain, it'll switch to the long chain, acyl-CoA dehydrogenase. When you split off a few more carbons, they get down to a 12 carbon. You switch over to a medium chain acyl-CoA dehydrogenase. And then when you get down to like the 8 or 6 carbon, you're going to switch to the short chain acyl-CoA dehydrogenase. Don't worry about the exact sizes of the fatty acids. Just know that there's actually 4 different acyl-CoA dehydrogenases depending on how long the fatty acid is. And the shorter it gets, you'll basically switch to the other one. And you'll keep switching until the whole thing is cut down. But there's actually 4 specific for short, medium, long, or very long chain fatty acids. As the fatty acid chain is shortened, it transfers it to the enzyme that acts on the shorter chain. And here's the written out reactions. And I guess after you guys have been through glycolysis and gluconeogenesis, I feel like this should be easy because this is it. This is 1 through 4. And you need to know 4 enzymes, and I'm going to tell you the ones that are probably the most important. And one of those is going to be the first step. The first step's always important. The first step, you have the acyl-CoA dehydrogenase. Actually, let me make sure. We're done with the no regulated enzymes here. You don't need to learn any regulated enzymes for beta oxidation. Just keep that note in your head or on your notes. This is the one pathway that we've already been through the regulated enzymes. The first regulated enzyme was hormone-sensitive lipase. The second regulated enzyme was the carnitine acyl transferase 1. Those are the 2 regulated enzymes, but there's no technically a regulated enzyme for beta oxidation itself. So acyl-CoA dehydrogenase is going to make an FADH2. The fatty acid is oxidized, and a double bond is going to be introduced between the alpha and beta bond carbons, and that's actually where it's going to be split. So step 1, acyl-CoA dehydrogenase is going to oxidize. It's going to make a double bond, and it's going to make an FADH2. The next step is a hydration step, which means we're going to add water, and that's all we do. We add water, and it's going to result in a hydroxyl group. This is probably one of those steps that I might not care about too much. Let's put it that way. The second step is also important because we make something. We make what's another dehydrogenase, and in this case, we're making an NADH. So it's actually called hydroxyl acyl-CoA dehydrogenase. And that's because it's working on a hydroxyl group, so that's why it's called a hydroxy acyl-CoA. And we make an NADH. So the third step, we make an NADH. The first step, we make an FADH2. The third step, we're making an NADH. And the fourth step, we're making an acetyl-CoA. We're making an acetyl-CoA in the fourth step. And that's going to be by an enzyme. I usually just call it a thiolase. Technically, the name is ketoacyl-CoA thiolase. That's the final step, and it makes the acetyl-CoA. And so each round of beta oxidation results in 1 acetyl-CoA, 1 NADH, and 1 FADH2. And you already know what we do with those. What do we do with them? What do we do with acetyl-CoA? We put it in the what cycle? The Krebs, TCA, whatever you want to call it, we put it in the Krebs cycle. And what do we do with NADH and FADH2? We put them where? In the electron transport chain to make ATP. And you already learned those last term, so you don't have to learn them again. But that's what's going to happen to them. And here it is, first step, right here. What's that? Removes 2 carbons from the fatty acyl chain each round. So each round removes 2 carbons at a time. Step 1 is the acyl-CoA dehydrogenase, and we make an FADH2. What we're doing here is introducing a double bond. That's going to be really important in a second here, so I'm pointing that out. The second step, and by the way, these are a little off-centered. Second step is here. The second step is where we add water, and we basically are going to convert this to a hydroxyl group. So it's going to go from a double bond to a hydroxyl group. And then the third step, so that was step 1, step 2, The third step is here, and another dehydrogenase, and we're gonna make another NADH. The last step is thiolysis. This is where we're going to split this guy, and so we end up with an acetyl-CoA and the rest of the fatty acid chain. So this fatty acid chain, all it's going to do is go back up here and start all over again. Acetyl-CoA, on the other hand, is going to go to the Krebs cycle. And we're going to keep doing this until we've broken that thing down all the way to the last 2 carbons. So if I had an 18 carbon fatty acid, how many acetyl-CoAs can I make from that 18 carbon? You guys still listening?
Yes.
What's 18 divided by 2? 9. You're going to make 9 acetyl-CoAs from an 18 carbon fatty acid. You're going to make 6 acetyl-CoAs from 12. Thank you. What are you going to make from palmitate? How many acetyl-CoAs can you make from palmitate? 8, because palmitate is a 16 carbon, is the most common one used. So you're going to make 8 acetyl-CoAs from that. So that's why you can make a lot of ATP from fatty acids. There's a lot more carbs are not going to have as much. Fatty acids are in a more reduced state, and so that's because they have a lot more carbons. Essentially you have like a glucose, remember, is a 6 carbon, whereas a fatty acid is upwards of 16 to 20 or more carbons. So you can actually have a lot more electrons to remove from a fatty acid than you do from glucose. And those electrons are what's important. Those electrons are going to be able to be used to make ATP from the electron transport chain. And remember the byproducts of the electron transport chain, or sorry, not the electron transport chain, but just these energy pathways in general, all those carbons, when you put them in the Krebs cycle, are going to be lost as carbon dioxide. And then when we get to the TCA cycle, oxygen is the final electron acceptor, and when it takes those electrons, you make water. You don't have to remember all this. I'm just reminding you for the boards that you're going to be taking sooner probably than you want to. But here's what I do want you to know. Palmitate is 16 carbons. If you do this many, many times, you can make 8 acetyl-CoAs. You can make upwards of 130 ATP from palmitate. How many ATPs can you make from glucose? 36. So 36 ATPs from glucose, 129 from palmitate. And this actually the textbook shows you exactly where you make them, but you don't have to memorize all that. So glucose, you can make upwards of 38 tops, but usually the textbooks say 36. It depends on the math that they do. But a 16 carbon can make upwards of 129. Yeah. No. Medium chain as well. So you could use an 8 carbon to do beta oxidation and make 4 acetyl-CoAs from an 8 carbon. It's usually palmitate, so it's usually a 16 carbon palmitate. But someone who, especially if they have malabsorption issues or fat issues, they are supplemented with medium chain. It supplements people. Athletes are starting to supplement with medium chain. Medium chain are actually oxidized better, more effectively than long chain. So people who have more medium, it's just we tend to have more long chain in our diet. But if you do tend to have more medium chain in your diet, then you'll be burning those by beta oxidation instead. Yeah. So this tells you, I can actually do the calculation and sit there and tell you how many ATPs, and this is an abbreviated form of that. So remember from FADH2, you can make around 2 ATP, and from NADPH, you can make around 3 ATP. And so this is kind of the math, but don't memorize it. I won't be asking you the math. This is what I want you to remember. You can do the math, but it's approximately 130 ATP from palmitate versus around 36 to 38 from glucose. But here I did all the math for you, or some of the math. But I will not be expecting you to regurgitate this math on the exam. But if you do want to know it, we can talk. All right. So this is from your textbook, enzymes involved in beta oxidation. So you have, so just the same enzymes here. You have the acyl-CoA dehydrogenase. You have the hydratase, which adds the water. You have another dehydrogenase where we make the NADH, and here's the thiolase where we make the acetyl-CoA. So this is the same figure. It's just from your textbook instead of the one that I used. I don't want to go back to it. And this is where your textbook did the math for you. So I had my own math. This is the textbook doing the math. But again, the whole point of this slide as well is to show that there's approximately 130 ATP that you can make using palmitate or palmitoyl-CoA. When you add a CoA to palmitate, you're supposed to call it palmitoyl-CoA. And this one is what I want to make sure I remember we get to before a couple things. Yeah. Saturated versus unsaturated fatty acids. So remember, you can use either for beta oxidation, but an unsaturated fatty acid will not make as much ATP or not make as much as acetyl-CoA. So you need one less fatty acyl-CoA every double bond. So for every double bond, and remember unsaturated are the ones that have the double bonds, every time you have a double bond, that means that you don't have to do that first step. So remember the first step is where you make a double bond between carbons. If you have an unsaturated fatty acid, you already have a double bond. And so you do not produce that first FADH2. Whenever there's already a double bond, you don't make that FADH2, which means you're not going to make as much energy or as much ATP from an unsaturated fatty acid than you will from a saturated fatty acid. So everyone, I want that to be something you remember. When you have an unsaturated fatty acid, it already has that double bond in it, so you make one less FADH2 everywhere there is already a double bond. So the key takeaway is that you make less energy, less ATP, because you make less FADH2 from an unsaturated fatty acid than a saturated fatty acid. Results in less energy because it's already less reduced. Remember, whenever there's a double bond, then you have less carbons or less hydrogens. And remember, when you're following electrons, you're kind of following these hydrogens. Whereas if you don't have a double bond, you have more hydrogens. You have a hydrogen here, you have a hydrogen here. On both sides of the carbon, you have a hydrogen. So you have more electrons to take away from it. But when you have a double bond here, you don't have that option. All right, does that kind of make sense?
Yes.
Because, well, I'm going to say this one more slide. Odd chain. So we talked about unsaturated, now odd chain. What happens when you get to the bottom of that chain and you're left with 3 carbons instead of a 2-carbon acetyl-CoA? That's essentially propionyl-CoA. So with odd chain fatty acids, you end up with propionyl-CoA, which is not a waste product. We can turn it into succinyl-CoA. And succinyl-CoA is an intermediate in the TCA cycle. So even if you're using unsaturated or odd chain, you can still use them, but it's a slightly different than using a saturated fatty acid. All right, guys, I have a really important meeting. I'm trying to make it by 3. I don't know if I'm going to. So I'm going to end it there and run out the door.
All right.